Hello, my name is Joshua Mugabi. I welcome you to Biology King. And in this session, we are going to discuss about membrane structure. Draw the structure of phospholipid and identify its properties. The phospholipid has got a phosphate, which is hydrophilic. It has also the fatty acids, which are hydrophobic. Phospholipid consists of the phosphate head, which is hydrophilic, and it is a water-loving. The phospholipid also consists of two fatty acids. Those two fatty acids are hydrophobic. That is, they are water-hating. The phosphate group is connected to the fatty acid tails via a glycerol group. Explain how the properties of phospholipids helps to maintain the structure of cell membranes. The phospholipids form a phospholipid bilayer. The hydrophilic phosphate heads face outwards into the aqueous solution. The hydrophobic fatty acid tails face inwards and are held in place by weak hydrophobic associations. Because the associations are weak, phospholipids are able to move within the bilayers. Identify and describe the two main models of membrane structure based on the diagrams below. The diagrams below, we have one. This is the Davison-Daniels model. The second one is the Nicholson-Singer model. The Davison-Daniel layer has got two layers of proteins, flanked a central phospholipid layer. Whereas the Nicholson-Singer, the transmembrane proteins are embedded within the phospholipid layer. Davison-Daniels model is also referred to as lipoprotein sandwich. Nicholson-Singer, this model is also referred to as the fluid mosaic model. Describe how the following electron micrograph supports the structure of Daniel model. The micrograph shows a trilaminar or 3 layers structures, 2 dark outer layers and a light inner layer. This supports the idea of 2 layers of proteins, that is the dark part, flanking a central phospholipid layer. outline the evidence that lead to the falsification of Danielli model and acceptance of Singer model. Now, not all membranes have a constant ratio of lipid to proteins. 2. Membrane proteins vary in size and are insoluble in water. 3. Membrane proteins are mobile and are not fixed in place. 4. Fracturing the membrane reveals a rough and irregular internal surface. All these ones are evidence that lead to falsification of Daniel's model and acceptance of Singer model. Explain what is meant by the term membrane fluidity. Membrane fluidity means that the bilayer is not static, and membrane components such as proteins can move positions. That is, they allow membranes to change shape and to break and to reform. Outline the role of cholesterol in animal cell membranes. Cholesterol reduces the membrane fluidity. That is, it mobilizes phospholipids at higher temperatures. Secondly, it reduces the membrane permeability to hydrophilic ions. It prevents crystallization. That is, it increases flexibility at lower temperatures. It also helps secure peripheral proteins by forming high-density lipid rafts capable of anchoring the protein. Differentiate between integral and peripheral membrane proteins. Integral proteins are permanently attached to the membrane and typically transmembrane. They span across the bilayer and may contain hydrophilic pores to mediate material transfer. 2. Peripheral proteins are temporarily attached by non-covalent interactions and associate with only one surface of the membrane. List the functions of membrane proteins. Junctions. These serve to connect and join two cells together. Enzymes. Fixing to membranes localize metabolic pathways. Transport. Responsible for facilitated diffusion and active transport. Recognition. May function as markers for cellular identification. Anchoring. Attachment points for cytoskeleton and extracellular matrix. Transduction. Function as receptors for peptide hormones. Label the following diagram of a plasma membrane. 1. Those are cholesterol in animal cell membrane. 2. The integral protein, or what we refer to as transmembrane. 3. Peripheral proteins. 4. Glycoprotein or receptor molecule. And 5 is the phospholipid, part of the bilayer. Hello friends, thank you very much for participating in Biology Kings. I encourage you to subscribe. Thank you.